And thank everyone for joining. And today what I'm going to cover is specifically focusing on support within dkNET 3 for enhancing rigor and reproducibility. So first, first thing that I wanted to do was for people who are new to dkNET, just give a one slide overview of what dkNET is, and then another slide just talking about some of the services, tools we provide. So dkNET was launched to serve needs of basic and clinical investigators in diabetes, digestive, endocrine, metabolic, kidney, urologic diseases, the focus of NIDDK at NIH. And we provide seamless access to a collection of diverse research resources. So we tie in to external databases, external data sources, information resources, information on materials, tools, funding opportunities. We also provide services on top of the literature, and I'll show you. And these are all things that will come out in the webinar today. And again, the focus of today is really the tools and services in support of rigor and reproducibility. And this is built around the resource identifiers. And so that's really going to be, the focus of today's webinar. dkNET 3.0 is our most recent release. So the final public release occurred earlier this month. There are number of areas of the portal that in terms of tools we provide. You'll see that there's tools regarding resource reports for various types of resources like cells and antibodies, and authentication reports for antibodies and cell lines. And so these are sort of the two main components I'll be focusing on today. There's also the discovery portal. So this is the sort of one of the core components of the portal is where we aggregate information from hundreds of different biomedical databases. Some of this information is used to power the resource reports and authentication reports. And then there's also the Hypothesis Center. So this is an area where we are partnering with other projects such as the Signaling Pathways Project to provide ways for users to easily explore in this initial case with the Signaling Pathways Project omics data, to generate or potentially test potential research hypotheses. So again, this webinar we're going to focus sort of on the left side, of the tools that we have, and the issues of rigor and reproducibility have really sort of taken the front line in the last number of years. And so, there's also issues in terms of, the economics, of rigor and reproducibility. So low reproducibility rates, within science, have issues both on the the scientific results side of things, but also in the sense of the funding that went into studies that might not have been properly constructed, or are using cell lines, antibodies that have issues, and therefore have issues, and in the resulting results that are published. So looking at, just the cost of this, there was a paper from Friedman 2017, looking at, issues, with irreproducible research and, the cost of this in terms of the billions of dollars. So, issues with biological reagents, in itself is, estimated $10.2 billion. And so that's a lot of money that would be better spent, if there were tools to assist researchers, in providing assistance, when studies are constructed. NIH has provided now a number of new grant guidelines, to help sort of improve Rigor and Reproducibility. There are a number of resources on NIH's website regarding this. And we also, within dkNET, within our portal sections, do provide links out to these various areas, so you can get information through dkNET, and then we also provide you then the links out to some of the background material from NIH. And there are new grant review criteria, right? So this is now filtering into what is needed for researchers in terms of preparing new proposals. And there's sort of 4 areas that NIH is requiring more explicit attention for applicants and reviewers. And there's a scientific premise, scientific rigor, consideration of relevant biological variables such as sex, and then the authentication of key biological and/or chemical resources. And so this is the area where dkNET has been looking at providing additional tools and additional support for researchers, for better information about the resources that they are using. And so, what is required, is that all grants must include a 1-page attachment informing the reviewers how grantees plan to authenticate, their key biological and chemical research resources. And this includes the cell lines, antibodies, organisms, specialty chemicals, and so on. So, this is now an integral part of the proposal process. But one of the big issues, with authenticating, but also verifying, that antibodies, cell lines, are acting, in a good way, that there are no problems with them, is being able to identify, in already published papers, what biological resources have been used. And a number of years back, we ran a pilot study. So this was published in 2013. Is that basically 50% of resources are not identifiable in the published literature. So trying to provide a baseline of information, to help researchers, choose more appropriate resources to help with, information on, validation information that's available, or information on problems, with resources. That's, very difficult to do if one can't even just identify the resources that are being used in scientific studies. And in many cases, in the paper, there's something that's written, very difficult. You can't find it. If you go back to, the community resource where, the organism or the antibody, was purchased, you can't go back and search and find that information. But the authors know, what was actually used. And so, it's just one example where, we had such a conversation with an author where we were trying to identify something in a paper, and then the author could point us directly to the resource and what was used. And, the idea then is, well, how can we, handle this? So one can imagine, that, what we show here is a little identifier that can point then directly to that information, for example, in this case, at the Jackson Laboratory. So looking across, various types of resources, how common is this? So again, this is from the Vasilevsky paper in 2013, where we look at, what is the fraction of resources that can be identified in the published literature, without the use of some type of other identifying mechanism. And when you look at this, you'll see that antibodies, cell lines, constructs, very poor, under 50%. Model organisms a bit better, at around 80%. But can we improve this? And of course, we can improve identifiability of resources if we include a unique identifier, right? So if each resource had a unique identifier, it'd be very easy to link those then to the actual resources in the source repositories. And here again, we show an example of a method section where we have these resource identifiers, one highlighted in orange here, for each of the key resources used. And this is similar to, how we, basically identify papers. So each paper, if you notice down below in the references, every paper has a digital object identifier. So again, an identifier that is then easily resolved. You can take that identifier and then you can go and easily find the paper and link directly to that paper using these identifiers. And so the resource identifiers, which I sort of previewed a bit there, were the result of the Resource Identification Initiative. And this was something that was organized through Force 11, the Future of Research Communication and Ease Scholarship. So this is a community of researchers, librarians, publishers, who are involved in a number of initiatives around e-scholarship. And the Resource Identification Initiative was this community-led effort to introduce better reporting standards for these research resources. And they needed to be search-friendly, uniform across publishers, and outside a paywall. And so uniform across publishers, one thing that publishers have had experience with is accession numbers. And so the RRIDs, these identifiers, were made to fit within how publishers have already been used to identifying items. For example, data in GenBank, is one example, for accession numbers that journals were familiar with. In addition, the project took advantage of NIH's already prior investment into community databases. So the model organism community, has a number of databases, that provide information on various model organisms, that we need to refer to through a number of other projects. There was an antibody, so through the Neuroscience Information Framework, the Antibody Registry, is a registry of antibodies that is used, both commercial and produced by labs and submitted. And through NIDDK and NIF-funded activities, we're able to use these aggregated database to create a resource identification portal. And dkNET really was the project that was able to provide sort of the pipelines and services, to pull all this together, to enable sort of the RRID initiative to take off. And again, these research resource identifiers, they're unique identifiers for these research resources. And the point I want to make, again, is that these are provided by independent community registries who track a single type of resource. So again, we're relying on these registries in the community, to provide information about the identifiers. And so the RRID initiative put together the standard format of the identifier, and this allows, as new types of resources come into the initiative, we're able to work with community repositories, these community registries, then to allow them to handle sort of the registration of new resources, and that they then work within the overall RRID sets of tools and services that are being implemented by us via dkNET, but also in terms of publishers. As publishers add more tooling to support RRIDs, they need to be able to support, their publication pipeline, and the incorporation of these RRIDs as well. So once we have, RRIDs embedded in paper, so we had an initial pilot project, and looking, at a future paper published by Bandrowski et al. In 2015, there's a marked improvement in the, identifiability, of antibodies, organisms, and tools, for these, for these resources. So again, having these identifiers, is a critical foundational layer, to provide services, in support of rigor reproducibility and the ability to get information on these resources for researchers to make more informed decisions. And this is being supported by major publishers. So Nature is one of the latest journals to come on board. But there are a number of large publishers who are requiring RRIDs. Other publishers are suggesting, are asking researchers to provide them. To provide this unique persistent identification for the tracking of these resources. And for example, when looking at Cell Press, they have introduced their STAR methods format, and so the STAR methods format includes, the RRID identifiers as part of that. And eLife itself is supporting the initiative, and they've also been one of the journals who has been tying in to some of the RRID services, that are provided by dkNET, to help with, with their validation of RRIDs as they're being published. The American Association for Cancer Research, has also information on resource identifiers, and again, so there are different tacks as to how journals are using this, but there is now a significant growth, in the use of RRIDs and in the number of publications that are using those. So why, I hinted at this before, but why, are we so, keen on having these resources identified, in papers? One is for tracking. So, who used a specific organism, who has used an antibody in the past? Can I find the papers related to that, so that I can see, what people have done, or any other information on the use of those antibodies. The second is, for rigor and transparency. So, for authenticating antibodies and cell lines, has there been, an issue with that cell line? Has it, is it known to be contaminated? Are there issues with an antibody? Some of the community repositories have some of this information, and so if these cell lines, antibodies are identified appropriately, it makes it really easy, to retrieve that type of information. So again, with these RRIDs, we have now the ability to have these resources identified, and with one click, you can go from the identifier, from the journal to the actual information page on that resource. And so within dkNET, we've been building up tool suites around these resource identifiers. The first is the resource reports, and so this is where we aggregate information about these research resources based on their RRIDs. Again, who is using them? Do they have issues? And also, is anyone near me using them? So, if you're struggling with a certain antibody and you don't know why, is there someone near me that has prior experience with this antibody? And can I contact them? And so the summary reports, that we produce for these resources are accessible through the front page of dkNET. You can go to the resource reports section and select what you're interested in. In this case, someone might be selecting interested in antibodies. You're taken to a search page where you can provide information to help narrow down the list of antibodies. For the different resource types, we provide special search boxes. So for example, for antibodies, we also have specific search boxes for vendor and catalog number, which provide an easier way to sort of hone in on the antibody that you're interested in case you have that information. And once you've done your search, you'll get back a Google-like result of the different items. And so what you'll notice is that in addition to basic information about the antibody, we also highlight if for example this resource was mentioned in the literature, and where do we know if it's been mentioned in the literature. And in this case, we know that this antibody has been mentioned more than 170 times in the resource literature. There's also a table view that one can switch to. So if you're looking at a way to sort of see this in a more tabular form, or also download this as a CSV spreadsheet, you're able to do that. And again, here we provide the information in tabular view. We also provide some word clouds, to help, for example, give you an idea of sort of the target organisms, for example, for an antibody that you can then easily review. So the summary report, once one selects, in this case, an antibody, we provide a summary report that has, again, information on the antibody or other resource, the number of times, again, it was cited in the literature. We provide, on this page, some of the most recent articles that we have found that use that antibody. But if you actually were to click on the full usage report, you'll be provided with an actual graph, of when we have found articles, that mention this antibody. We also provide information on other research resources that are frequently mentioned, in this case, with this antibody. So this is sort of information on co-mentions. For antibodies, we also do that for software tools, which is actually really interesting in some cases where you might be using a certain analytic tool, and then the co-mentions, in many cases, provide information on other analytic tools, that people have used in conjunction with that, and also different viewers, for example. So you might be analyzing your data using one tool, but there might be additional viewing tools, that you can utilize to visualize your data. And so these are things that we can pull out with these co-mentions. We also have information on additional sort of report filters and mentions. So one of them, as I mentioned, was the Collaborators Network. And here what we're doing is, with the data from PubMed for certain journals and PubMed entries, we have information on the institutions for the authors themselves. And so we can actually, make that also searchable. And so you can find collaborators near you. This is one of the newest pieces to the information network, and is an area where we're adding, more location information monthly. So this is something where we're sort of growing our amount of data that we have around these researcher locations. One of the other things that we are able to actually aggregate within these resource reports are ratings and alerts. So I talked a little bit about sort of the discovery data aggregation system, that exists within dkNET, and using that, we're able to aggregate information, from a number of sites, ENCODE, the Human Protein Atlas, resource centers, such as the Intestinal Stem Cells Consortium, and we're able to then aggregate that information, do some processing of that, and then provide information, for, an antibody. Has it been discontinued? Is there a problematic cell line? Do we have validation data available at the Human Protein Atlas? And so, aggregating that information and making it easier for researchers to find that information is one of the key things that are part of the resource reports. But this also ties into the authentication reports, which I'll get into in a couple of slides. So one of the important things is, if I have a resource that I don't find, so, is it an antibody that you've developed in your lab or a cell line that you've developed? For registering these resources, again, we rely on these community resources. So if you have an antibody, you can go to the antibody registry page. We provide links to all these. And, at the antibody registry, you can actually register your antibody. And then, once that's been, curated, we then are able to, federate that information, and then that becomes part of, our data that we then provide through these resource reports and other reports. We also, provide information, contacting cell source for the organ model organisms. There are a number of different community registries that you might need to contact, so we provide a list of those. One of the other things that we've been working towards in dkNET is also increasing sort of the number of resources, that are identifiable. So one of the most recent ones that we've added is biosamples, and so this was collaborative work with the Integrated Islet Distribution Program and the HERN program, which is funded by NIDDK. And here we worked with the biosamples databases in Europe, and also at NCBI at NIH here in the US to provide identifiers for pancreatic islets used by researchers. And in developing this with NCBI, and the IADP, we made sure to ensure that that it isn't just, for example, islets that can be registered this way, but that basically any group that wants to register biosamples with NCBI Biosample and obtain these SAM identifiers can be used to generate research resource identifiers that can then be used in publications. And so again, looking towards being able to identify additional resources, we can add additional community authorities can be brought in support of resource identification, and so this is another area that we've been working on. So now it's sort of turning focus to the authentication reports, that I mentioned earlier on in terms of what NIH is requesting in terms of proposal submissions. And this really gets to issues with, do we, publish, papers with, do we still publish papers with contaminated cell lines, right? And so there is, a lot of issues in terms of, knowing within these publications, if there is, again, gets back to the identifiability, do we know if, a certain cell line in a paper, or antibody or other resource, has had issues or problems, or, has some documentation related to it being problematic. Within cell lines, one of the sort of cornerstones for this information is sort of a curated database that's put together by the International Cell Line Authentication Committee. And so this is the ICLAC, in terms of, authenticating cell lines, has a number of guidelines in terms of what should be done. But again, one of the key ones is check, if it is a known contaminated cell line. And so ICLAC is one of the groups that contributes to Cellosaurus, which is the community authority for cell lines that's used within the RRID ecosystem. And, a recent paper published in 2019 by Babak et al., looked at papers that either did not include resource identifiers to identify their cell lines, and those that did. So what we actually, what this paper actually discovered was that, when using resource identifiers, there are less, problematic cell lines in those sets of papers. And so, is this something that, as researchers, when they're publishing their paper, they're looking at the research, the RRIDs, through RRIDs, they find out that there is an issue with a cell line. Does that cause, a difference in terms of how they publish or go back and do additional validation on the cell lines that they used. And so this is, using RRID sort of post-publication. But what we really want to be able to do is do this type of checking sort of before we get to the publication stage. And the same issues, with antibodies. So are we publishing papers with antibodies, that have issues with cross-reactivity, are being used in the wrong application? And so, these warnings, are critical, so we have, information on problematic cell lines, that we can pull out. And through, the systems that we have within dkNET, we can combine that information also with information that's provided to assist researchers in preparing these authentication plans for cell lines and antibodies. And so within dkNET, we have also incorporated information, for example, in this case, validation pillars, for the validation of antibodies. And so this is, a proposal that was in Nature Methods from Ulin et al. And, this was an international working group that came together to develop these validation pillars. And then this is information that we provide through our authentication report section within dkNET. And so when one gets to the authentication report section on the left here of the screen, you can see that there is, a bunch of information related to the basic authentication information. And so, the cell line authentication plan information, the antibody authentication plan information, basically provides this information, for example, about the antibody validation pillars, and also, the information from ICLAC, in terms of validation of cell lines, in terms of general information. However, what we are, since we do have these resource identifiers and we are aggregating information about these, we can actually provide researchers with a custom authentication report. And this includes then the basic resource information that I covered earlier, but also then aggregates all this information about the noted issues or problems, with the resources in a single report. And so when one goes, when you've opened a dkNET account, you can go and create a report. We then provide the ability to find and add your resources, and right away, as you're doing this, the system is checking the items that you are adding. So we provide information as you start collecting the various resources that you plan on using in an experiment. We're providing the information that we have about this. And so in this case, just to sort of highlight this, we've selected a number of problematic resources. So when you're searching for these, you can then you're immediately notified that there are potential issues with that resource. Once you actually have these items added into your authentication report, you can actually then go create a report, and from that you can download a PDF, which you can use to copy and paste information. You can also copy and paste information from the website itself. And so what we provide is information on the cell lines themselves. So if you look on the left and you see that with the table of contents, first we provide a table for the cell lines that you've provided with some basic information, including any potential alerts that we might have about that resource. We then have information about authentication plan, in terms of what is recommended, and then we provide the references that provide all this information. So really trying to help the researchers in terms of providing some of this basic information for their use. And the same thing for antibodies. So here again, the antibody section is very similar. We have the identification of the antibodies, again, this listing of the antibodies. We then provide a validation information. And here again, we point to, in this case, the validation pillars that I covered beforehand. Providing all that information and the references down below, for researchers. At the end of the authentication report, we have a resource index. And again, this resource index has all the information that we have retrieved from the external community authority about that cell line or antibody, and we provide that then in a listing format at the end of these reports. And so again, through dkNET's ability to sort of collate a lot of this information about resources, from these community authorities, we're able to put together these, two very valuable services, the resource reports themselves, and then the ability to, for researchers to, bring this information together in authentication reports, that they can use for proposals when they're planning, on doing a study. So, again, RRIDs, first, utilized more on the publication end. So after you've done your study, you, when looking for a research resource identifier for your resource, might be notified, about, problems or other uses, of a resource. But in this case, with the authentication reports, we're providing a tool that allows people and researchers, when they're constructing a study, to initially collect information regarding, those antibody cell lines right up front, to see, if there is anything that has been found, or, just to get information on, other researchers who have used this antibody, so that it gives you the ability to go into the literature more easily, to find documented uses, of that antibody or cell line. And then, as your study is progressing, you actually can go back to the authentication reports and retrieve up-to-date information. So one of the things that wanted to point out is that we have the ability to preview these reports at multiple time points. So you can actually create a report initially, generate a PDF of that report, and then come back 3 months later, 6 months later, and see if there's any additional information that we have aggregated about those resources, as your study is ongoing. So it also provides a way, for you to track, about any additional information, about those resources that you might be using in your experiments. So again, this is a sort of a brief overview of these dkNET features in terms of supporting more rigor and transparency around the use of resources. Another area that is very important in terms of reproducibility is documentation of protocols. And so the next webinar is actually going to be one of our first external webinars where we'll be bringing in protocols.io to give more information about their system of protocol documentation. And so this webinar about reproducibility, the methods behind the data, will be given by protocols.io on Friday, June 21st at the same time. And so I welcome you to attend that webinar, because protocols.io is a really good resource for being able to document not just your own protocols internally, and to be able to modify them and version and track sort of your protocols, but also for publishing these full protocols to the research community. So that covers, I think, what I wanted to discuss today on this webinar. And so if there are any questions, I'd be happy to take them now.
Jeff, hi, this is John Cadiz. Regarding your ratings and alerts, provided that the resource is in dkNET, would there be an API way of pulling those alerts data? We've been talking in Hearn about some way to try to leverage what you're doing with that, so that we can inform our community for the Hearn resources when you guys find a problem with something.
Yes. So I didn't didn't cover the sort of the programmer side of things in this webinar, but yes, we do have a set of APIs that can be used to retrieve this data. And so that's documented within our API section. So when you get an API key, via dkNET, we also have the documentation for the services listed there. And we can also, provide you with sort of samples, in terms of how you would retrieve that information.
Thank you, Jeff. Very nice work with you and your team.
Hi, Jeff. There is a question from the chat box. Can you walk us through development of a typical rigor and reproducibility report in real time from beginning to the output?
So let's see. Hold on. Do I have a browser? Hold on. Let me get out of this presentation. Again, going into demo mode, so let's see if my computer doesn't revolt on me. let's see, sorry. Get through my password wallet there. Okay, so with the authentication reports, you would go to the authentication report section. And here again, we have the cell line basic information on the left, along with the antibody information. Again, the information on, for the sources for authentication information are all given in this section as well, in case you're just looking for basic background information. But if you do want to create an authentication report, you can, start your report. There are a number of steps to do this. So, first you need to create a new report. So let's do this webinar, May 24, demo for webinar. So I can submit that. And so then I get to the authentication report dashboard page for this specific report. And then I can, search, for, let me just search for something, so GFAP. The list then shows up below. And what you'll notice is that, we have the table view. I mentioned the resource report. So this little icon in the table will actually take you to a resource report for that antibody in this case, and so basically we can go in, I can add that to my report. You'll notice that I get the green confirmation that it has been successfully added. It shows up in my report items on top. I can go cell line as well, and so here I can add a cell line, and if I want to find problematic cell line, I can just for the demo, I can add a cell line as well. And in this case, you'll notice that the warning message comes up for this. So basically, you go through the resources that you're adding in terms of antibodies and cell lines to this report. You continue to add them. They continue to show up in the report items listing. When you're done, you can actually then go to preview the report, right? So that's the next step. So I click to preview the report, and then I get the actual report preview that I walked you through earlier. Again, notice that the alerts, we try to make these as visible as possible, also in the reports, and then gives you that information in terms of the authentication plans for cell lines, the references, the validation pillars for antibodies. Additionally, for model organisms, since we do have information aggregated about that, we can also link out to information that we might have on specific knockout organisms that might help you in your validation, and then provides the resource index down below. And the last thing is that I can save a PDF of this report. So if I create a PDF, this creates a read-only copy, in the sense that the PDF is a snapshot of this report. So if I go back to the report, you'll notice that underneath the preview report button, which I clicked before, there's now a list of PDF reports that I generated. So, again, as you go through, sort of constructing a study and then being engaged in the research for that study, you can always go back, and basically take snapshots, of the information that we have on these resources. And again, we are trying to aggregate information from a number of different resources. We may not have information from every resource out there. If there is another resource that provides validation information or information about problematic resources, that we don't have yet, please let us know. We'll be more than happy to, try and aggregate more information for these reports. So that's basically the basic walkthrough. I don't know if that was covered everything that you wanted to, but I think that's the main areas of the report. Any other questions?
I don't see other question on this.
Okay.
Yeah. Mm-hmm.
Again, and again, this is the first release of dkNET 3, and so really these are two new components in terms of what we're releasing to the scientific community, and we want your feedback. So if there's anything additional you'd like to see in the resource reports or in the authentication reports, don't hesitate to let us know. We're trying to make these useful tools for the research community to help improve the rigor and reproducibility of ongoing research. So don't hesitate to get into contact with us. Okay, and so seeing no other questions, I can give everyone back 7 minutes to their day. So hope everyone joins us for our next webinar. I think it's going to be a really good one, protocols.io. And so see everyone June 21st.